DNA Replication by Leah, Teresa, Emma, and Christina. There are 4 nitrogenous bases involved in replication. These bases can be purines, which have 2 carbon-nitrogen bases, or pyrimidines, which have 1 carbon-nitrogen bases. Adenine and cytosine are purines, while thymine and guanine are pyrimidines. Purines can only bond to pyrimidines. Adenine bonds to thymine with 2 hydrogen bonds, and guanine bonds to cytosine with 3 hydrogen bonds. These are known as base pairs, and this rule was discovered by Erwin Chargaff. A person's base sequence is what makes them different. There are about 6 billion base pairs in every cell. Mutations are changes in the original sequence. Mismatches in base pairing or damage from toxins are the most common causes of mutations. However, they are extremely rare. The strands of DNA molecules are considered sugar-phosphate chains as they are composed of phosphate and ribose, nitrogenous bases attached to the ribosome of the DNA strand. The replication process begins during the S phase at specific spots called origins of replication, or ORIs. The enzyme helicase enters at the origin of replication site and unwinds the double helix into 2 single-strand DNA molecules. Helicase breaks the hydrogen bonds holding these bases DNA together, A with T, C with G, and forms a replication bubble with the replication fork moving in 2 directions. Single-strand binding proteins bind to the DNA strands and keep them elongated because they have the tendency to rejoin or bind to the outside of the strand. In order to break these bonds, helicase requires energy. Therefore, the ATP created throughout cellular respiration fuels this process. For the leading strand, RNA primase attaches to RNA nucleotides on the DNA strand to form an RNA primer so DNA replication can be initiated. The enzyme DNA polymerase then replaces the primer and adds nucleotides to create base pairs. This occurs continuously, forming a daughter strand by adding DNA nucleotides in the 3' to 5' direction of the parent strand. After this process, the nucleotide replacement enzyme replaces the initial RNA nucleotides with DNA nucleotides. The parent and daughter strands are considered antiparallel because they have opposite alignments. One runs from 3' to 5' and the other from 5' to 3'. Please note that although not depicted in the video, the daughter strand of the leading strand runs from 5' to 3' because it is antiparallel to the parent strand. Unfortunately, DNA polymerase can only copy strands in the 3', 5' direction, and the lagging daughter strand lies in the 5', 3' direction. So RNA primase attaches to RNA nucleotides to form RNA primers, which initiates replication in chunks known as Okazaki fragments. The enzyme works backwards along the strand. Finally, DNA ligase stitches the fragments together. DNA replication is semi-conservative, meaning that each end in the DNA double helix acts as a template for the synthesis of a new complementary strand. The process goes from 1 starting molecule to 2 final molecules, with each newly formed double helixes containing 1 new and 1 old strand. Because DNA replication is anabolic and new molecules are being produced, this process is considered an endergonic reaction, one that requires energy. The circulatory system, respiratory system, and digestive system each play a critical role in DNA replication. The circulatory system is responsible for transporting the oxygen taken in by the respiratory system to the cells of the body in order to allow cellular respiration. Oxygen is required for the aerobic cell respiration processes of the Krebs cycle and the electron transport chain. Respiratory system is required because it allows us to take in oxygen in addition to form ATP and other products of cellular respiration. Humans must breathe out carbon dioxide. We must digest carbohydrates to extract glucose. Glucose is the initial reactant of cellular respiration, which creates the ATP that drives DNA replication.